In this video, we look at two types of white blood cells that make up a large part of what's called your secondary immunity. So your ability of your body to fight stuff off when it gets inside. So these two white blood cells are phagocytes and lymphocytes. So phagocytes, lymphocytes. In this video, it looks like we're going to look at lymphocytes. So all of the cells in your body have markers on their surface. They are self-markers. They tell your body that that cell is from your body. Cells that are not from your body have markers on the outside that are not deliberately designed to tell your body that they're not from you, but if your body doesn't recognize them, it assumes they are foreign or not yours. So when a lymphocyte comes across a microbe that it doesn't recognize, it makes lots and lots of copies of itself. This is what's called clonal selection if you go up to higher, but for us it just makes copies of itself. So the first thing it does, recognizes that it doesn't know what that is, makes lots of copies of itself, so it's got friends to back it up, and each copy of the lymphocyte can make something called an antibody against that foreign microbe. So what is an antibody and how are they made? Here we go. So a virus gains access to the body and multiplies inside your cells. So viruses are actually incapable of living outside a host cell. So on the outside of the virus, there are little proteins called markers. Some viral particles become attached to by their markers to a type of lymphocyte that recognizes them as foreign. So while these viruses are going between your cells, trying to replicate themselves, a lymphocyte might come across them. If it looks at the outside of the virus and it says, wait a minute, those aren't my proteins, that's foreign, it will start to panic. The lymphocyte makes more copies of itself, and each copy is able to produce antibodies. Now, the interesting thing is, each lymphocyte is specific for only one little protein on the outside of a virus. So the protein just around the corner from that here would need a completely different type of lymphocyte to recognize it. So because lymphocytes are specific to just one little protein on the outside, they make very specific antibodies as well. So the antibodies join onto the invading particle and cause them to clump together, so they can't infect any more of your cells. They're later engulfed by what's called a phagocyte. So to follow that through, the virus gets into your body, but it has little proteins on the outside that your body can recognize do not belong to you. The lymphocyte comes along, recognizes that, and makes lots of copies of itself. The lymphocyte is specific for only that protein on the outside of the virus. The lymphocyte's made lots of copies of itself, and it's able to then produce what are called antibodies, which are also able to attach onto the virus at that little protein that the lymphocyte recognized. It doesn't just attach to one, it's got two arms, so it can attach to this one, but it can also attach to the one beside it. And by chaining together, it makes this big, big clump of antibodies and antigen complexes, it's called, which stop the viruses being able to be all fast and speedy and get in and out your cells, and makes them easier for your body to destroy with what's called phagocyte. So the antibodies, as I've said, are specific to only one type of protein on the microbe. For this reason, we say they're specific. So if you look at the shape here, pointy, pointy, circly, circly, squarey, squarey, which one of these would fit onto that microbe? So it'd be C, if you think about it, because that's the one that has the curved bit. You can imagine that, not quite the right shape, but you can imagine a bigger version of that fitting onto the outside of this and attaching this microbe to another one so it can't get away. So this is just a little summary of what you need to know. So a lymphocyte recognizes the microbe as foreign, the lymphocyte reproduces and makes lots of copies of itself. Each of these lymphocytes can make hundreds and hundreds of antibodies that are identical to each other. The antibodies join with the microbe to cause them to clump together, and something called a phagocyte comes along and eats the clump of microbes. That is what you want to write on page 27 of your booklets. So what we're going to look at now is phagocytes. So the lessons are usually taught in the opposite way around, but it doesn't matter in the slightest. So by the end of this, you're going to be able to tell me about phagocytes and how they fight off microbes. So, when microbes infect the body, the white blood cell number increases. We've talked about this. It's the word I use, clonal selection, but it's the lymphocytes making lots of copies of themselves. And more blood will go to the site of the infection. The large number of white blood cells at the infection site will speed up the destruction of that organism and helps you to heal. So in this part of the video, we're looking at phagocytes. So phagocytes, if the skin gets damaged, microbes can enter the bloodstream and make us ill. The white blood cells called phagocytes will notice anything foreign and strange in exactly the same way that the lymphocytes do as well. If they notice something is wrong, they are the first proper line of white blood cell defense. So what they do is they go up to them and they eat them. It's a process called phagocytosis. So first stage, a phagocyte moves towards the pathogen. It takes in the pathogen. So you can see it's kind of doing what's called invagination. It's going around the outside of this pathogen, completely covering it and joining up at the other side. So the pathogen is now stuck inside in this little bubble, which we call a vesicle. The phagocyte, now it's got it trapped inside it, can digest the pathogen, so the bad thing, the virus or bacteria, using enzymes. So it puts lots of nasty things called enzymes into this little bit here, and it digests the pathogen. So they respond in the same way to all the different microbes in the body. So they're a fairly non-specific way to do this. So on page 26, this is why you tend to do them the opposite way around. It asks you to draw an annotated diagram. You don't necessarily need to do that. If you just write down these words in the right order, that's okay. So a pathogen moves towards the phagocyte moves towards the pathogen. It surrounds the pathogen and takes it into itself. The pathogen becomes digested and is now harmless. So you could try and make a little model of this at home if you want. You could combine what you know about lymphocytes with what you know about phagocytes. If you want to send me a picture, that'd be absolutely lovely. So that is everything you need to know for this section. Hopefully that makes sense. If not, please do ask.